This is a look at Greenland. Okay, Arctic people have occupied Greenland for 5,000 years. The earliest inhabitants were the Sakic people, who occupied tiny settlements on Greenland's coast from between 4,800 to 2,500 years ago. The mystery of their origin was solved in 2010 when geneticists determined that the entire DNA sequence of a 4,000-year-old Sakic male, nicknamed Inuk, whose remains were recovered from an archaeological site on the western coast of Greenland, um, matched data that they had traced back all the way to Eastern Europe. Okay? Scientists extracted the DNA from hair tufts found in the permafrost, and by comparing the DNA with sequences from known population, the geneticists were able to demonstrate that the Sakic people are most closely related to the Chukuki, which are a present-day group of indigenous people from Russia. This finding indicates that the Sakic people originated from hunters who trekked from Siberia eastward across Alaska and Canada to Greenland, arriving in the New World independently of the groups that gave rise to the Native Americans and the Inuit, who populate the Arctic regions. So this is all done through sequence similarity and comparison, or what we call homology. Okay, and this is part of what allows us to do things like draw um, evolutionary trees. Okay, genetic material, which didn't have an identity until relatively uh, recently when compared to our knowledge of heritable characteristics, okay, has to satisfy the following criteria, right? It has to contain complex information. It must be capable of storing large amounts of information in a tiny volume, which provide the instructions and the traits and the functions of an organism. Next, the genetic material has to replicate faithfully. Every organism begins life as a single cell, and to produce a complex multicellular organism, the single cell has to undergo billions of cell divisions, and at each division, genetic instructions have to be accurately transmitted to descendant cells, and when the organisms reproduce and pass their genes to their progeny, the genetic instructions must be copied with fidelity, meaning accurately. Next, the genetic material has to encode the phenotype. The genetic material must have the capacity to be expressed as a phenotype, i.e., to code for traits. The product of the gene is often a protein or an RNA molecule, so there must always be a mechanism for genetic instructions in the DNA to be copied into RNAs and in many cases into protein. Finally, the genetic material must have the capacity to change. Genetic information must have the ability to vary because different species and individual members of the same species differ in their genetic makeup. Okay? Back in 1868, Johann Miescher graduated from medical school in Switzerland, and he was influenced by an uncle who believed that the key to understanding disease lay in the chemistry of tissues. Miescher traveled to Germany to study under Ernst Felix Hoppe Seyler, an early leader in the emerging field of biochemistry. Under his direction, Miescher turned his attention to the chemistry of pus, a substance of medical importance. Pus contains white blood cells, which have large nuclei, and Miescher developed a method for isolating the nuclei. The tiny amounts of nuclear material that he obtained were insufficient for a thorough chemical analysis, but he did establish that the nuclear material contained a novel substance that was slightly acidic and high in phosphorus. This material, which we now know as DNA and protein, he called nuclein. The substance was later renamed nucleic acid by one of his students. In the 1800s, Kossel carried out further work on the chemistry of DNA and determined that it contains four nitrogenous bases: adenine, cytosine, guanine, and thymine, and we abbreviate those A, C, G, and T. As additional studies of the chemistry of DNA were completed in the 40s and 50s, the notion of DNA as a simple invariant molecule began to change. Chargaff and his colleagues carefully measured the amounts of four bases in DNA from a variety of organisms, and they found that DNA from different organisms varies greatly in base composition. This finding disproved what used to be called the tetranucleotide hypothesis. Chargaff discovered that within each species there is some regularity in the ratios of bases. The amount of adenine is always equal to the amount of thymine, and the amount of guanine is always equal to the amount of cytosine. These findings became known as the Chargaff rules. However, the cause of these ratios between the bases was unknown at the time. We didn't yet understand the three-dimensional structure of DNA, and that's something that would come a bit later. We're going to talk about some landmark experiments that established nucleic acid as the genetic material. We'll address the Griffith experiment, the MacLeod and McCarthy experiment, and finally we'll address the Hershey-Chase. An initial step in identifying DNA as the source of genetic information came with the discovery of a phenomenon known as transformation. This was first observed in 1928 by Griffith, who was an English physician whose special interest was the bacteria that causes pneumonia, known as Streptococcus pneumoniae. Griffith has succeeded in isolating several different strains of this bacteria, types 1, 2, and 3, and so on. In the virulent forms of the strain, each bacteria is surrounded by a polysaccharide coat, which makes the bacterial colony appear smooth when grown on an agar plate. He also found that the virulent forms occasionally mutated to non-virulent, which lack a polysaccharide coat and produce a rough-appearing colony. We designate these two phenotypes Cap-S and Cap-R. Griffith observed that small amounts of living type 3S bacteria injected into mice caused the mice to develop pneumonia and die, and when he examined the dead mice, he found large amounts of 3S bacteria in the blood. When Griffith injected 2R bacteria into mice, the mice lived and no bacteria were recovered from their blood. Griffith knew that boiling killed all bacteria and destroyed their virulence. When he injected large amounts of heat-killed 3S bacteria into mice, the mice lived and no 3S bacteria were recovered from their blood. The results of these experiments were not unusual. However, Griffith got a surprise when he injected his mice with small amounts of living 2R bacteria along with large amounts of heat-killed 3S. Because both type 2R bacteria and heat-killed 3S were non-virulent, he expected the mice to live, but 5 days after the injection, the mice developed pneumonia and died. When Griffith examined the blood from the hearts of these mice, he observed that live 3S bacteria were present. In addition, these bacteria retained their 3S characteristics through several generations. Their virulence was therefore heritable, i.e. it was passed down as a trait. At the time of Griffith's report, Avery was a microbiologist at the Rockefeller Institute. At first, Avery was skeptical of Griffith's results, but after other microbiologists repeated those results with other bacteria, Avery set out to try and understand the nature of the transforming material. After a decade of research, Avery, Colin MacLeod, and Macklin McCarty succeeded in isolating and partially purifying the transforming substance. And they showed that it had a chemical composition closely matching that of DNA and different from that of protein. Enzymes such as trypsin and chymotrypsin, known to break down protein, didn't have an effect on the transforming substance. Ribonuclease, which destroys RNA, had no effect, but enzymes that destroy DNA eliminated the activity of the transforming substance. Avery, MacLeod, and McCarthy showed that the transforming substance precipitated at approximately the same rate as purified DNA and that it absorbed UV light at the same wavelength as DNA. These results were published in 1944 and provided evidence that the transforming principle, and therefore genetic information, was in DNA. However, new theories in science are rarely accepted right away, and many biologists continue to prefer the hypothesis that the genetic material was protein. Now, there were reasons for this. Okay, protein was more complex, and as a result was thought to have the capability of containing more information. Okay, there was a bias against a chemically simple molecule with relatively few building blocks as being an information repository. That's one of the reasons that it took a while for DNA to be accepted as the transforming material. A second piece of evidence that indicated DNA was a genetic material resulted from a study of the T2 bacteriophage that was utilized by Hershey and Chase. The T2 phage is a virus that infects bacteria that being E. coli. As we saw earlier, a phage reproduces by attaching to the outer wall of a bacterial cell, injecting its genetic material, which then replicates and directs the cell to synthesize phage protein. The phage DNA becomes encapsulated in the phage protein and generates progeny phage that can rupture the cell. At the time of the Hershey-Chase study, biologists didn't understand exactly how phages reproduce. What they did know was that the T2 phage is about 50% protein and 50% DNA, and that a phage infects a bacterial cell first by attaching to the cell wall, and that progeny phages are ultimately produced inside the cell. Because the progeny carry the same traits as the infecting phage, genetic material from the infecting phage has to be transmitted to the progeny, but how this genetic transmission took place wasn't understood. Hershey and Chase designed a series of experiments to determine whether protein or DNA transmitted in phage reproduction was the transforming agent, or the heritable agent, let's say. To follow the fates of protein and DNA, they used radioactive phosphorus and sulfur. The phosphorus can radioactively label genetic material, DNA, and the sulfur radioactively labels protein. Okay, so their experiment follows. All right, so let's take a look at it.
In the Hershey-Chase experiment, bacterial viruses, called phage, were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing S35 radioactively labeled amino acids. This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacteria. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The phage produced in these cells contained no radioactivity. Vigorous shaking caused the empty protein coats to be removed, but did not interfere with production of new phage in the cell. In the second part of the experiment, phage were produced in a medium containing P32 labeled deoxyribonucleotides. This resulted in a phage population with P32 labeled DNA, but no radioactive label in the protein. When the phage infected the bacteria, the P32 labeled DNA entered the cell and could be found in phage subsequently produced in the infected bacteria. This demonstrated that the DNA, but not the protein, carries the genetic information for a new generation of phage.
Before Watson and Crick's breakthrough in elucidating the structure of DNA, a lot of the basic chemistry of DNA had already been determined by Miescher, Kossel, Levine, and Chargaff, as well as others who established that DNA is made up of nucleotides and that each nucleotide contains a sugar, a nitrogenous base, and a phosphate group. But how the nucleotides fit together in three-dimensional structure was unclear. In 1947, Asbury began studying the three-dimensional structure of DNA using X-ray diffraction, as shown here. X-rays beamed at a molecule are reflected in a specific pattern, and this reveals aspects of the structure of the molecule. However, this production of diffraction images didn't provide enough resolution to reveal the structure. A research group at King's College in London, led by Wilkins, also used X-ray diffraction to study DNA. Working in Wilkins' lab, Rosalind Franklin observed strikingly better images of the molecule, but Wilkins and Franklin's progress in developing a complete structure of the molecule was slowed by the basically the animus between the two researchers. So X-ray diffraction is simply a method for analyzing the three-dimensional shape and structure of a chemical in which crystals of the substance are bombarded with X-rays, which hit the crystals, bounce off, and produce a pattern of spots on a detector. The pattern of the spots produced on the detector provides information about the molecular structure. What's critical in doing this is that you have to get pure crystals of the material that you're analyzing, and that took some time. Okay, this is one of the reasons. This is one of the ways, for instance, today that we're able to elucidate the structure of proteins is through things like X-ray diffraction, generating protein crystals. Okay. In most organisms, DNA carries genetic information, but a few viruses use RNA and not DNA as their material. This was demonstrated in '56 by Conrad and Singer, who worked with tobacco mosaic virus, which infects and causes disease in tobacco plants. This virus has a single molecule of RNA surrounded by a helically arranged cylinder of protein. These researchers found that after separating the RNA and the protein in TMB, he could mix the RNA and protein of different strains of TMB and get intact infectious viral particles. These researchers then created hybrid viruses by mixing RNA and protein from different strains of TMB. When these hybrid viruses infected tobacco leaves, new viral particles were observed. The new viral progeny were identical with the strain from which the RNA had been isolated and did not exhibit the characteristics of the strain that donated the protein. These results show that RNA carries the genetic information in TMB. Okay, so viruses are an exception. Viruses basically are delivery vehicles for genetic material, and they're not technically living things. So what they do is they utilize a host in order to reproduce, and it's the host machinery that gets hijacked to create the new virus. Okay, viral genetic material can be double or single-stranded DNA, double or single-stranded RNA. Okay, and there's a variety of enzymatic processes, some of which are unique to the virus, that allow it to utilize information other than double-stranded DNA as genetic material. In addition, in '56, Girer and Schramm demonstrated that RNA isolated in tobacco mosaic virus is sufficient to infect tobacco plants and produce new viral particles, and this confirmed that RNA carries genetic instructions in the virus. So basically what they did was they transformed plant cells with just the genetic material, and lo and behold, that genetic material was sufficient to start up viral production in healthy cells from healthy plant tissue. So the building blocks of DNA, which are nucleotides, have three components. They have a sugar, a phosphate group, and a base. Okay, structure will be shown here on subsequent pages. The purine or pyrimidine base gets its name from the fact that it's an organic molecule that contains nitrogen, and organic molecules that contain nitrogen are bases because of the lone pair of electrons on the nitrogen, which can scavenge protons from the surrounding water. Okay, and so that's the definition of a base, something that has the ability to reduce the proton concentration. DNA is a long molecule, and it therefore is termed a macromolecule. It's also a polymer. As an example, within each human chromosome, a single DNA molecule, if stretched out straight, would be several centimeters long, thousands of times longer than the cell. In spite of its large size, DNA is a simple structure. It's a polymer made up of repeating units called nucleotides. The repeating units of DNA have a sugar, a phosphate group, and a nitrogen-containing base. The sugars of nucleic acids, which are called pentose sugars because they have 5 carbons, these carbons numbered 1' through 5', indicate the positions on the ring. The sugars of DNA and RNA are a little bit different in structure in that RNA sugar, called ribose, has a hydroxyl group attached to the 2' carbon, whereas deoxyribose has simply a hydrogen. And this is one of the reasons that ribose is much less stable in solution than deoxyribose is, okay? Because the hydroxyl group in ribose can catalyze a chemical reaction within the molecule that breaks apart the polymer, whereas in deoxyribose, that hydroxyl group is missing. Okay? This provides specific functions for each nucleic acid, right? These nucleotides bear in their nitrogenous bases information, okay? Furthermore, additional oxygen atom in the RNA molecule makes it more reactive and less chemically stable, as we already pointed out, and for this reason, DNA is better suited to be the long-term storage molecule for information. So deoxyribose is the 5-carbon sugar in DNA lacking the hydroxyl group at the 2' carbon, while ribose has the 2' carbon. Okay? The second component of a nucleotide is the nitrogenous base, which can be either of two types, a purine or pyrimidine. Each purine has a 6-member ring attached to a 5-member ring, where pyrimidine has a 6-member ring only. Both DNA and RNA have two purines, adenine and guanine, which differ in the positions of their double bonds and in the groups attached to the 6-member ring. 3 pyrimidines are common in nucleic acids, cytosine, thymine, and uracil. Cytosine and thymine are present in, cytosine and guanine are present in RNA and DNA, as is adenine. Thymine is found only in DNA, and uracil is found only in RNA. The 3 pyrimidines differ in the groups of atoms attached to the carbon of the ring and in the number of double bonds. In a nucleotide, the nitrogenous bases always form a covalent bond with the 1' carbon atom of the sugar. A deoxyribose or ribose sugar and base are referred to as the nucleoside. Okay? So the nucleoside is the sugar and the base. The nucleotide is the phosphate, the sugar, and the base. Okay? Remember that thymine is a pyrimidine base in DNA but not RNA. Guanine, a purine in DNA and RNA. Adenine, a purine in DNA and RNA. Pyrimidines are a type of nitrogenous base in DNA and RNA. Cytosine, thymine, and uracil are all pyrimidines. Purine is a type of nitrogenous base in DNA and RNA. Adenine and guanine are the purines. And then a nitrogenous base is just a nitrogen-containing base that's one of the 3 parts of the nucleotide. Okay, so you can see here that you've got nitrogenous bases that are relatively large and some that are relatively small. Okay, and we're going to find out that in the double-stranded structure of DNA that you always have a purine matched up with a pyrimidine because of spacing considerations in the middle of the DNA double helix. We should also note this is a base, okay? The nitrogen atoms have free pairs of electrons on them that can scavenge protons, and that's why it's called a base. This is where the information is contained. It's the sequence of the nucleotides that dictates the genetic information, okay? So just like letters of the alphabet, you can write almost any major novel or newspaper using the alphabet and arranging the words in different formations, okay? And the same is true with DNA, right? These are the pieces of information. There's only 4 letters in the DNA alphabet, and you can arrange them in any order and any length, and as a result generate infinite amounts of information, practically. We should also note that these rings are flat because of the conjugated double bonds, so these are flat like a frisbee, okay? And we should also note the presence of electronegative atoms. Nitrogen and oxygen here have the ability to pull electrons towards them. The oxygens will tend to have a negative charge associated with them. The nitrogens will be electron-rich as well, okay? This results in hydrogen bonding across the double helix, okay, where the hydrogen atoms that are present on purines or pyrimidines can also form associations with oxygen atoms or with other nitrogen atoms on the neighboring base on the opposite strand. Okay, so when we get to the structure of DNA, you'll see what that's about. The third component of a nucleotide is its phosphate group, which is made up of a phosphorus atom bound to 4 oxygens. Phosphate groups are found in every nucleotide and frequently carry a negative charge, and that makes the DNA acidic. Because again, what will happen here is that the hydrogen will be attracted to the negative oxygen, form a hydroxyl group. That causes the proton concentration in the solution to drop, okay? Whereas if we were to take a phosphate group that had the hydroxyl groups present, it would easily ionize and donate the protons to the surrounding water, okay, and that would cause the pH to go down, okay. Okay. Phosphate groups cause the DNA to be acidic. The phosphate group is always bound to the 5' carbon atom of the sugar in a nucleotide. Okay, so you can see the structure here. All right. DNA nucleotides are properly known as deoxyribonucleotides or deoxyribonucleoside 5' monophosphates because there's 4 kinds of bases. There are 4 different kinds of DNA nucleotides. The equivalent RNA nucleotides are termed ribonucleotides or ribonucleotide 5' monophosphates. RNA molecules sometimes have additional rare bases which are modified forms of their 4 common bases. The modified bases will be talked about a little bit later. The names for DNA bases, nucleotides, and nucleosides will be shown in a table that we're going to pop up here in a minute. Okay, so ribonucleotide is the basic building block of RNA, has a ribose, a phosphate, and a nitrogenous base, and the deoxyribonucleotide is the counterpart in DNA. Okay, so notice, right? This will be in common, as will this, with every nucleotide. The only thing that changes is this, right? So this is where the information is located. This is where the information is. Okay? All right. Oh, and by the way, we should point out that although the rings are drawn as if they're flat here on the sugars, they are not, okay? They have sometimes a chair shape to them where the oxygen may sit up or sit down. And that's because every carbon indicated here at the bend or explicitly drawn has a tetrahedral shape to it when it's bound to 4 different chemical groups, okay? So just giving you a sort of a setup for, you know, the 3-dimensional structure of the whole thing, right? And again, these are flat like pancakes, okay, sitting out here. So there are the base symbols, the nucleotide symbol, the nucleoside, and the nucleoside symbol. Okay? The secondary structure of DNA is that of a double helix, which is a backbone formed through phosphodiester bonds consisting of hydrogen bonding and base pairing, and it has an anti-parallel structure. Okay, so DNA is made up of many nucleotides connected by covalent bonds, which join the 5' phosphate group of one nucleotide to the 3' hydroxyl of another. Note that the structures shown here are flattened into 2 dimensions, although the molecule itself is 3-dimensional. These bonds, called phosphodiesters, are strong covalent bonds, and a series of nucleotides linked in this way is a polynucleotide strand. The backbone of the strand is composed of alternating sugars and phosphate groups. The bases project away from the long axis of the strand. The negative charges of the phosphate groups are frequently neutralized by their association with positive charges on proteins or metals or other molecules. So a polynucleotide strand is simply a series of nucleotides linked together by phosphodiester bonds, and a phosphodiester linkage is a strong covalent bond that joins the 5' phosphate group of one nucleotide to the 3' hydroxyl of the next in the strand. A critical characteristic of polynucleotide strands are their directions or polarity. At one end of the strand, a free phosphate called the 5' phosphate marks the beginning of the strand, whereas at the 3' end we have a 3' hydroxyl group which marks the end of the strand. Okay? So when we talk about DNA being anti-parallel, one strand will run in the 5' to 3' direction and the other in the opposite direction. Okay? Still 5' to 3', but oriented in the exact reverse orientation. Okay? So remember, these nucleotides, they have shape to them, right? They have ends to them. They're not symmetrical on all sides, okay? So we stack these building blocks in one direction here, 5' to 3', and they only pair with the opposite strand when they're stacked the way they're indicated here, okay? And that allows the base pairing in the core of the molecule to occur, okay? Whereas in RNA, we're talking usually about a single-stranded molecule, okay? So let's take a look at DNA and RNA.
The structure of the DNA molecule was first inferred by James Watson and Francis Crick, based primarily on X-ray crystallography data collected by Maurice Wilkins and Rosalind Franklin. and chemical analysis of base composition of DNA conducted by Erwin Chargaff. The key features of the structure are its right-handed double helical structure. Each helix consists of an alternating sugar-phosphate backbone with nitrogen bases projecting toward the interior of each helix. One complete 360-degree turn of the helix covers 10 bases of length and equals 3.4 nanometers in physical distance along the axis of the molecule. The width of the double helix is 2 nanometers. The nucleotide bases are attached inside each backbone of the molecule so that the nucleotides in one helix or strand are hydrogen bonded to the bases in the other helix or strand. The hydrogen bonds hold the two strands of the double helix together. An adenine in one strand is always paired with a thymine in the other strand, and a guanine in one strand is always paired with a cytosine in the other strand. Note that guanine-cytosine base pairs form 3 hydrogen bonds, while adenine-thymine base pairs form 2 hydrogen bonds. This makes guanine-cytosine base pairs more stable than adenine-thymine base pairs. Nucleotide pairing between strands also allows the sequence in one strand to determine the sequence in the complementary strand. The two ends of a strand are not identical. Note that on one end of each strand, a 3' hydroxyl group of the deoxyribose sugar is not involved in the backbone, or we say it's free, while at the other end of the same strand, the 5' hydroxyl group of the deoxyribose sugar at the end is free or may contain a phosphate that is free and not bonded to another deoxyribose sugar. This dissimilarity of the two ends of a strand creates the ability to uniquely distinguish each end of the strand. Because of this polarity of each strand, we can see that the two strands of DNA are oriented in opposite directions, or we say that they are antiparallel. In three-dimensional space, the two helices are not equally spaced along the axis of the molecule such that a major groove and a minor groove are created by the asymmetric spacing of the antiparallel helix backbones. The major groove and minor groove play important roles in the binding of proteins that regulate gene transcription.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The two polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
So as we indicated earlier, right, DNA has both a three-dimensional and a two-dimensional structure. The secondary structure of DNA is made up of the double helix, okay, and it was identified by Watson and Crick, and it's commonly referred to as bDNA, as we saw in the video earlier. And there are other secondary structures that can take place as well. As we've seen, DNA normally consists of two polynucleotide strands that are antiparallel and complementary. The exceptions are single-stranded DNA molecules that are in some viruses. The precise three-dimensional shape of the molecule can change depending on the conditions in which the DNA exists. The 3D structure of DNA described by Watson and Crick is called the bDNA structure, and this exists when plenty of water surrounds the molecule so it's adequately hydrated, and there are no unusual base sequences in the DNA, conditions that are usually present in the nuclei of cells. bDNA structure is the most stable configuration for a random sequence of nucleotides under physiological conditions, and most evidence suggests this is what most of the DNA looks like. So bDNA is the right-handed helical structure of DNA that exists when water is abundant. The secondary structure described by Watson and Crick in a 2-page paper in Nature, probably the most common DNA structure in all cells, right? So saying that bDNA has a right-handed helix means that it moves in a clockwise spiral. There's about 10 base pairs per 360-degree rotation in the helix, so each pair is twisted 36 degrees relative to the base next to it. The base pairs are 0.34 nanometers apart, so each complete rotation of the molecule consists of about 3.4 nanometers. The diameter of the helix is 2 nanometers, and the bases are perpendicular to the long axis of the DNA molecule. A space-filling molecule shows that bDNA has a slim and elongated structure. The spiraling of the nucleotide strands creates major and minor grooves, as we saw earlier in the video. And these are features important for the binding of proteins that regulate the expression of genetic information. Okay, so you can see again the different structures, right? The space-filling model and this more two-dimensional graphic representation of the DNA. The core of the DNA tends to be water poor. Okay, there's relatively few water molecules in here. The backbone of the DNA tends to associate very readily with water. Okay, so just be aware of that as well. Although hydrogen bonding takes place in the core here. Okay. Another secondary structure that DNA can assume is aDNA. This exists if less water is around. Like bDNA, aDNA is a right-handed helix, but it's shorter and wider than bDNA, and its bases are tilted away from the main axis of the molecule. aDNA has been detected in some DNA protein complexes and in the spores of certain bacteria. So aDNA is a right-handed helical structure that exists in low water conditions. A different secondary structure called zDNA forms a left-handed helix. In this structure, the sugar phosphate backbone zigzags back and forth and produces a pattern similar to its name, right? A zDNA structure can result if the molecule contains particular base sequences, such as stretches of alternating C and G nucleotides. Scientists have found that zDNA-specific antibodies bind to regions of DNA that are being transcribed into RNA, and that suggests that zDNA may play a role in gene expression. Additional secondary structures of DNA, such as cDNA and ddNA, can form under special lab conditions or in DNA with particular base sequences. So zDNA is just a secondary structure of DNA characterized by 12 base pairs per turn, a left-handed helix, and a sugar phosphate backbone that zigzags back and forth. Okay, so you can see that structure all the way on the right? So we call this the normal form, and this is the Z form and the A form. So when you look at most DNA, it's going to look like the middle space-filling diagram. Now, information gets passed through a cell in several ways, right? One way you're probably familiar with is translation, in which the information contained in an RNA molecule is used to generate a protein molecule. And this happens in the cytoplasm of both bacteria and eukaryotic cells, while transcription, which is a process where RNA is synthesized from a DNA template, happens in the nucleus of eukaryotic cells and in the cytoplasm of prokaryotic cells. Okay? So DNA replication, something that takes place in all living cells, right? RNA replication is used in some viruses. RNA is synthesized from an RNA template, in this case. A little sidebar here, that's how COVID-19 reproduces its genetic material. Well, reverse transcription is the production of DNA from an RNA template, and that involves a novel enzyme called reverse transcriptase that is part of the architecture of the virus itself. So it represents a unique function in the virus that we don't have in our normal cells, and thus can be a drug target to fight the virus. The central dogma of biology is the idea that genetic information runs from DNA to RNA to protein in a one-way path. Okay? Replication errors are just incorporated errors that are reproduced and it leads to mutation. Or replication is the process whereby DNA is reproduced, and in some viruses, RNA is reproduced. Okay? Now, there are exceptions to this information flow, DNA to RNA to protein, right? For instance, in a retrovirus, we have an RNA chunk of genetic material that gets copied into DNA, and then the DNA is used to transcribe RNA, and then that RNA is used to make protein. Okay, so in a retrovirus, it goes from RNA to DNA to RNA to protein. Okay, a little bit unusual. And then in some viruses, of course, we have just plain old RNA to RNA to protein. Okay, and then in some viruses, we have simply RNA to protein. All right, so there's all sorts of different pathways that can take place. Most of the unusual pathways are produced by viral reproduction, and again, we have to stress that viruses are not technically living pathogens. Some special structures that form in DNA and RNA include hairpin structures, in which a single strand of nucleotide can be complementary within the strand and form what we call a hairpin loop. Okay, if I were to draw a hairpin loop, it would look. Let's do it in blue. Okay. It would look like this, where let's say this is the 5' end of the molecule, right? And then we have the RNA sequence, and then we have this stem, and then we have a loop here, and we have another stem, and we have the rest of the sequence, and this would be the 3' end of the single-stranded molecule. And then the sequences are such that the 5' to 3' part of the stem, right, is complementary to the 3' to 5' part of the stem over here, okay? And that's how you're able to generate this type of structure, okay? When the complementary sequences are contiguous, the hairpin has a stem and no loop, and when they're not contiguous, there's going to be a loop in there, okay? So this would be a non-contiguous example, whereas a contiguous would look basically like this, okay? Bing, just like that. Where that's the 3' end and that's the 5' end, okay? And then the complementarity is such that we go like that and then like that, right? And those are complementary, okay? RNA molecules can contain numerous hairpins, and that allows them to fold up into complex structures, and that can sometimes be used as a way to allow certain enzymatic reactions to take place, okay? So both DNA and RNA can form these special structures. A hairpin consists of a region of paired bases, which forms a stem, and a region of unpaired bases between complementary sequences, which forms a loop at the end of the stem. In B there, you can see a stem with no loop, and in A, you can see a stem with a loop, okay? In C, you see the secondary structure of the RNA component of RNase P of E. coli. The RNA molecules often have complex secondary structure. Okay? Sequences within the single strand of nucleotides can be complementary to each other and form those hydrogen bonds and produce those double-stranded regions. The internal base pairing imparts secondary structure to the single-stranded molecule, and we have the example of the hairpin and the hairpin loop. Okay? When the complementary sequences are contiguous, we form the stem with no loop, and when they're not, we form a loop with a stem. Okay? Secondary structures are critical in functions of a lot of RNA molecules, as we'll see a little bit later. Okay? So the hairpin is just the secondary structure formed when sequences of nucleotide in the same strand are complementary and can pair with each other. Okay? It basically can buckle up. DNA sequences can also sometimes form three-stranded structures or triplex structures called hDNA. When some of the DNA unwinds and a single polynucleotide strand from one part of the molecule pairs with a double-strand DNA from another part of the molecule. This is possible because under certain conditions, one base can simultaneously pair with two other bases. hDNA often occurs in long sequences containing only purines or only pyrimidines. Some triplexes consist of one strand of purines paired with two strands of pyrimidines. Other triplexes consist of one strand of pyrimidines with two strands of purines. Sequences capable of adopting hDNA conformations are common in mammalian genomes, and evidence suggests that hDNA occurs under natural conditions. Recent research has demonstrated that hDNA breaks more readily than double-stranded, which leads to higher rates of mutation in regions where this happens. Quadruplex structures involving four strands of DNA can also occur under these conditions. So hDNA is just a structure consisting of three nucleotide strands, which can occur when a single nucleotide strand from one part of a DNA molecule pairs with a double strand from another. Okay, so you can see examples here, right? There's your triple DNA, right? And here is a close-up of the triple DNA, right? 1, 2, 3 strands, right? Notice the double complementarity, right? The A's here are pairing with the T's on either side, the C's, and so on, okay? Adenine and cytosine are normally methylated in bacteria. In eukaryotic DNA, cytosine bases are sometimes methylated to form something called 5-methylcytosine. The extent of the cytosine methylation is going to vary among different eukaryotes. In most animal cells, about 5% of the cytosine bases are methylated, but there's no methylation of cytosine in yeast, and 50% of the cytosine bases in plants are methylated. Why eukaryotic organisms differ so widely in methylation isn't really clear. So 5-methylcytosine is just a modified form of cytosine containing a methyl group at one of the ring positions. Now you might wonder, okay, so what? Why do we care? 5-methylcytosine can serve as a marker for the binding of certain proteins to DNA. Okay, 5-methylguanosine is frequently used as the cap in RNA molecules at the 5' end to indicate where the ribosome should begin binding and scanning for translation start sites. Okay, so you could think of them as like little signal flares or markers on the DNA. Okay, and the enzymes put them there. All right. Okay, I thank you for listening, and I'll see everybody in the next podcast.